food, drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info.
Thanks for having me, Nick.
Do you want to just start off by telling everyone a little bit about who you are and what your lab studies?
Yeah, sure. So my name's Sara. I'm an assistant professor in the Department of Metabolism and Nutritional Programming at the Andel Institute, which is a private research institution in Grand Rapids, Michigan. And I started the lab here in the summer of 2021, and we're focused on mitochondrial biology and mechanisms that regulate mitochondrial function. And right now we have a particular focus on the mitochondrial fatty acid synthesis pathway, which I'll probably refer to it a lot as mitoFAS for abbreviation, but it's a pathway in the mitochondrial matrix that builds fatty acids.
Yeah, and so normally, I mean, when you first learn about mitochondria in school, like grade school, and in general, when you hear about mitochondria, we're typically talking about these organelles in the context of breaking down food and using it to transform energy to create ATP, the currency of energy in our cells. So we think of them as sort of catabolic organelles. They're part of the process of breaking food down and then converting that into ATP, the stuff that sort of powers all of the stuff in our cells. But mitochondria also are anabolic. They build stuff. They do fatty acid synthesis, as we'll discuss. That's a lot of what you focus on. So we're going to talk about that, but I want to start off pretty high level for just a little while to give people a sense for what mitochondria are most well known for and some of the basics of their structure, because I think that'll help us understand the fatty acid synthesis side. So at a high level, can you just walk us through the basic process by which things like fats and carbs are broken down and used to transform energy into ATP? In particular, we all have an intuitive sense of like breaking down food and using it to make energy. But at the cellular level, for someone like you, what does broken down actually mean? What do these things actually get broken down into? And then what happens from there?
Yeah, absolutely. So you're totally right. We think of mitochondria as these breakdown organelles because that's their classic function. Everybody knows this phrase, mitochondria are the powerhouse of the cell, right? So what that really means to me is when you think of your cell as taking up fuel sources in the form of sugars or fats. So typically we think about glucose as the primary fuel that's a sugar in our cells. That goes through a series of chemical reactions in the cytoplasm that are going to make it into a molecule called pyruvate. And pyruvate is the molecule that actually is going to enter the mitochondria and then get further broken down to make that cellular energy substrate, ATP. So mitochondria are famous for being this double membrane organelle. They have an inner membrane and an outer membrane. The outer membrane is pretty permeable to most small molecules. And the inner membrane is not. It has a lot of active transport processes that bring specific metabolites into the mitochondrial matrix. So pyruvate is going to enter through a specific transporter called the mitochondrial pyruvate carrier. And then it's going to be acted on by another series of enzymes in a cycle that a lot of people have also heard about. It's called the TCA cycle or the citric acid cycle or the Krebs cycle. And the main job of that cycle is making reduced cofactor molecules. And those are NADH and FADH2. And what those cofactors are is they're really electron carriers. So those enzymes that are catalyzing the reactions in the TCA cycle, they're going to hand electrons off to these carrier molecules, NADH and FADH2, and those are then going to pass them to something called the electron transport chain, or the ETC. The electron transport chain is this big series of molecular machines that sit in that inner membrane. And they use the flow of those electrons to pump protons outside the mitochondrial matrix. It's almost like setting up a battery because what that does is it creates a separation of charge across the membrane. And then we can use that battery to let those protons flow back into the matrix. And that's what actually makes ATP through the last complex in the chain that's just known as ATP synthase.
Mm-hmm. So basically, we eat some food. We might eat a carbohydrate rich meal and maybe there's starch in there that'll get broken down into glucose. Maybe we eat a fatty meal. Those fats will be turned into smaller fatty acids. Whether we're talking about fats or carbs, the stuff that we're ingesting that we call food, that will ultimately get, those molecules will get turned into other molecules. And ultimately what's being created are molecules like FAD and NAD that are good at holding on to electrons. And we're sort of using them to move electrons through the inner membrane of the mitochondria. So the mitochondria have two membranes, not just the outer membrane like we kind of are used to thinking about cells, but then another one on the inside. And basically the electrons are basically passing from the food to these other molecules and through that membrane, kind of like a wire.
Yeah, you could totally think of it that way. Or, sometimes when I talk about this with family members, I'll invoke that old I Love Lucy episode where there's like the assembly line that's a conveyor belt of chocolates, and Lucy has to like wrap the chocolates and it goes faster and faster.
Mm-hmm.
And it's really sort of like that. It's like an assembly line of these metabolic reactions handing off electrons to these complexes, and they have to go down the chain to get to that final product that's the wrapped chocolate. But, if things can't keep up or go haywire, then, you end up sort of in this predicament. But anyway, that's another story.
Yeah. So there's this component here of chemical transformation, turning molecules from the food into other molecules that can hold electrons. There's this component of separating charge and allowing electron flow within the mitochondria. And even if people are unfamiliar with, the actual physics of a battery, we sort of all know, okay, there's a positive side, there's a negative side, there's this charge separation. You've got to, put the jumper cables on the right end of the battery, and the food is participating in this flow of electricity ultimately.
Yep, absolutely.
And so, okay, so you mentioned some terms, and I just want to maybe cover them again and paint people a visual picture. So the mitochondria have two membranes, an outer membrane and an inner membrane. And then you mentioned the mitochondrial matrix. Can you just sort of give us again a bird's eye view of what these sort of key areas in the mitochondria are?
Yeah, absolutely. So you covered it really well there. So you have this outer membrane that's permeable to a lot of small molecules. So metabolites can really move in and out. And then you have an inner membrane, and that's what's going to hold that electron transport chain and set up that separation of charge, that molecular battery that we're thinking about. And then the matrix is just what we call the inside of the inner membrane, like that fluid filled space that's inside the very inner part. And so that is an area that is going to not be membrane bound proteins, but more soluble things and reactions. A lot of the breakdown of the molecules is actually going to happen in that mitochondrial matrix or that very inner part.
Mm-hmm, mm-hmm. And so the electron transport chain is in the inner mitochondrial membrane. Again, very high level basics. We don't need to cover all the details, but what is this electron transport chain? What are its basic components, and what is it doing?
Yeah, so there are four main large protein complexes that make up the chain, and they are made of all these different protein subunits that are put together along with reactive cofactors. Like there are iron sulfur clusters in a lot of these complexes. There are hemes and reactive coppers. And so metals, a lot of the time, that you would think of as being able to catch those electrons and then pass them down a chain to the next metal ion in the complex. And then ultimately, the last complex, complex 4, is going to pass those electrons to molecular oxygen. And it will hold it until it gets two of those electrons and then it becomes water. And so the electron transport chain is literally the part of your cell that breathes because it consumes oxygen, because oxygen is what ultimately is going to accept those electrons when they're done moving down that chain.
Right. So literally, this is why we breathe. The oxygen is going into the mitochondria, and it's pulling the electrons through this whole apparatus.
Yep, absolutely. And
And
then
so,
at this,
yep, go
oh,
ahead.
I was just going to say at the same time that it's doing all of that, three of those complexes are pumping protons across that membrane. And so that's setting up that separation of charge between the matrix and the inner membrane space across that inner membrane. And it's that separation of charge across that inner membrane that's going to act like that battery potential and drive ATP formation.
Hmm. And then, again, at the beginning of here, we're going to stay pretty high level. But when we think about this whole apparatus, this electron transport chain on the inside of the mitochondria in the inner membrane, we're thinking about oxygen pulling electrons. Electrons are flowing. This is bioelectricity. We're creating this proton gradient. We're using this ultimately to turn the ATP ACE that generates ATP, the cellular energy for cells. What is the difference between, say, fats and carbs that people can start to think about here in terms of, like, are they ultimately broken down, and from the mitochondria's perspective they are identical because we're transforming them into the same stuff, or do the fats and the carbs kind of get used in different ways with respect to the ATP production, the electron flow, and how this whole apparatus is actually utilizing those food components?
Yeah, so I would say yes and no. At the end of the day, we do break them down into the same stuff. So I talked about pyruvate, and pyruvate being the thing downstream of sugar that or starches, carbs, that's actually going to enter the mitochondria. The next step in that process is an enzyme called pyruvate dehydrogenase, and it's going to break pyruvate down into a really important molecule called acetyl CoA. On the flip side, if you eat a lot of fats, you bring the fats into your cells, and then the fats are also actually able to enter into the mitochondria. Um, and they undergo a process called beta oxidation or fatty acid oxidation that's going to break them down into acetyl-CoA as well. So after you have acetyl-CoA, everything downstream is the same. Acetyl-CoA is going to enter that citric acid cycle. It's going to hand off its electrons to NADH, and it's going to power the ETC and make ATP.
Hmm.
But the difference is how much acetyl-CoA you get from each
Mm.
of those substrates and how you get there.
Mm.
So if you're talking about one molecule of glucose, like one single molecule of sugar, you're only going to get two molecules of pyruvate and two acetyl-CoAs produced.
Hmm.
But if you're talking about a single fatty acid like palmitate, palmitate is a fully saturated fatty acid that has 16 carbons in it. And so from that one molecule of fat, you can get 8 molecules of acetyl-CoA.
Got it. It's basically just a bigger molecule. There's more carbons there, and you can get more acetyl-CoA from that larger thing.
That's one part of it. The other part is that process of making the acetyl-CoA from the fat. It also produces some of those mitochondrial cofactors as well.
Hmm.
And so if you're doing beta oxidation, you actually feed some electrons into the electron transport chain before you ever get to acetyl-CoA too.
Hmm. Hmm. And what, like, how would you start to think about the ramifications of that? What would be sort of the difference in the whole apparatus here and how it's functioning if you're feeding in acetyl-CoA, period, versus feeding in acetyl-CoA after injecting some of these electrons into the electron transport chain?
Well, it just means that one molecule of fat is going to hold a whole lot more energy than one molecule of sugar.
Got it. Got it. Okay, so that's what ultimately, I mean, this is probably also why fat is, sort of, we think of that as the energy store in our bodies, because you can just store more per gram.
Yeah, absolutely.
Okay, so acetyl-CoA is sort of this common thing we get to no matter what we're eating. And what exactly is acetyl-CoA like at the molecular level? Is there something special about its structure, or how it functions that why is this sort of the common thing that gets made? What is special about it?
Well, I think it's really the simplest fatty acid is really what it is.
Hmm.
It's so those fats when they come into the mitochondria, they're also hooked to a CoA molecule as well. CoA is just another carrier, but instead of an electron carrier, you can think of it as a fatty acid carrier.
Hmm.
So acetyl-CoA is the simplest fat. It's a two-carbon fat on one of those CoA molecules. And CoA can donate it to a molecule called oxaloacetate so that it can enter that TCA cycle. So it's going to make a molecule called citrate. And so it's the common way to get to citrate in the mitochondria. And then citrate is also really special because it has multiple destinations or multiple places it can go in the cell. So it can enter the TCA cycle like we've been talking about, or it can also exit the mitochondria and go back to the cytoplasm where it can perform additional roles in the cell.
Mm-hmm, mm-hmm. Okay, so this has sort of been our very basic overview of what we might call the traditional function of mitochondria. This is the process by which we break down food and we're transforming energy to make ATP. I've covered this before on the podcast. There's obviously lots of other resources out there. The side of things that you focus on is sort of the non-traditional function, one of the non-traditional functions of mitochondria, and that's that they synthesize, they make stuff. They don't just break it down. They synthesize fatty acids. So I'll kind of let you drive a little bit in terms of the best way to hook into this side of things. But again, at a high level, why would the mitochondria, want or need, if I'm going to anthropomorphize, to build and make its own fatty acids?
Yeah, that's a great teleological question. And I don't think we 100% understand the answer because we know, right, that mitochondria, I just said they're taking up fatty acids
Right,
from
right.
other areas in the cell for beta oxidation. So it seems a little futile for them to also have a pathway to make their own. They also take up phospholipids from the endoplasmic reticulum. And so, yeah, I think that's something that my lab is actually really keen to understand is just why does this pathway exist?
Right,
But
right. But
it's orthology.
and even
Oh,
if we don't
go.
know why,
Yeah.
everything you're going to tell us, like there's a lot of resources that go into that ability being there. So it must be doing something because
Yeah,
it's there.
absolutely.
Yeah.
So, and it's very, very evolutionarily conserved as well. So,
Mm.
mitochondrial fatty acid synthesis looks a lot like bacterial fatty acid synthesis, which I think is not surprising if you know about the endosymbiotic origins of mitochondria, right? There's this hypothesis that mitochondria were a bacteria that an ancient eukaryotic ancestor ate and engulfed and then decided to keep around, right?
Hmm.
And so the pathway looks a lot like the fatty acid synthesis pathway in bacteria. And it builds fatty acids on a soluble acyl carrier protein. And when researchers were first studying this pathway, when they discovered it in like the late 80s and early 90s, the first product of the pathway that they found was this peculiar lipid called lipoic acid. So, lipoic acid is an 8 carbon lipid and it has the ability to make a disulfide bond. So, it's kind of special in that way. It can act as an antioxidant, or what it's really important for in cells is it's a cofactor for a bunch of mitochondrial enzymes.
Mm.
And so in the late 80s, early 90s, researchers found this function of the pathway and they were kind of like, cool, the pathway is important and it's been maintained throughout evolution because it makes lipoic acid and we can't scavenge lipoic acid. We have to make it de novo.
Mm.
And for some reason, we want to build our own fatty acids to feed that pathway, not any of the ones that we've taken up exogenously. And so that's what it does. Case closed, end of story. But of course, it's not that simple,
Right, right.
right?
Okay, so mitochondria can make these fatty acids. They can make lipoic acid. It doesn't appear that we can get it another way. We have to do this, it seems, even if we don't know all the details of why this is happening and what's going on. But lipoic acid, I had not heard of this. I know virtually nothing about it beyond what I learned in the last 30 minutes before this conversation. It's got disulfide bonds, so 2 sulfur atoms. Does that tell us anything? Disulfide bonds, this is something that comes up a lot in biochemistry. Can you just give us like a sense for what does that type of chemistry do? What is it good for?
Yeah, it basically means that it can participate in oxidation reduction reactions. And
Mm-hmm.
so it can, similar to those cofactors we were talking about earlier, in another way, it can accept electrons or donate electrons. So probably the most famous molecule that uses disulfides in a redox way like that is glutathione, which a lot
Mm-hmm.
of
Mm-hmm.
people have heard of.
Yeah, that's a major molecule people talk about in the context of antioxidant pathways. So, okay, these disulfide bonds, they're really good at oxidation reduction reactions. You also have mentioned a term a couple of times that I want to make sure people understand. So, what is a cofactor exactly?
Yeah, a cofactor is a small molecule that an enzyme is going to use to be able to catalyze whatever reaction it catalyzes. And so, a lot of enzymes, right? Enzymes are proteins that are going to catalyze a biochemical reaction. That means make the reaction go faster, lower the activation energy for the reaction. And so, um, a lot of proteins can do that just on their own using the amino acids and the chemistry that's available in the protein, but some can't do that. There's more chemistry that happens in biology than can just be catalyzed by those amino acids and the chemistry available. And so a lot of enzymes will use another small molecule that we call a cofactor, and they'll use the chemistry provided by that cofactor to do their catalytic mechanism.
I see. So enzymes participate in um, chemical transformations, so basically turning one thing into another, but they can't necessarily do that on their own, or at least not as well as they can when they've got these other cofactors with them or floating nearby. So they're kind of like helper molecules that basically help the chemical transformations happen at the appropriate rate.
Yeah, absolutely. And in the case of the enzymes that have lipoic acid, they really can't do the chemistry they need to do without it at all.
Hmm, hmm. Yeah, so if you don't have, that would imply that without lipoic acid, if something breaks in the ability of the mitochondria to make this, then stuff goes really wrong.
Yeah, absolutely. That's true.
So lipoic acid is a cofactor. So cofactors, like a lot of vitamins, right, are cofactors. That's kind of why we care about them as vitamins. So they're not the enzyme, they're not the protein doing the transformation, but they are very, very important in enabling that to happen or happen as well as it needs to happen. That's why we, want to get our vitamins and stuff. I've never really thought about fatty acids as cofactors before, but clearly they do function that way, or they can, some of them.
Yeah, yeah. There's a whole bunch of ways that you could, yeah, get from a fatty acid to several of the cofactors, and but lipoic acid is the one I'm most familiar with. They sell lipoic acid as a supplement in
Hmm.
fitness stores and other places, but I already told you that we have to make it. We can't
Right,
scavenge it, and
right.
so we think that any effects that individuals see when they take those supplements, they really likely just come from its antioxidant function and not its function as a membrane or as an enzyme cofactor because enzymes can't just grab it from the environment.
Got it. So to be used as a cofactor by the enzymes that use lipoic acid, it just has to come from the lipoic acid synthesized in our mitochondria. It can't be sucked in from the outside and used for that function. But supplemental lipoic acid does potentially still retain the antioxidant properties of lipoic acid.
Yep. I mean, it's a fatty acid, so it
Yeah.
can still go in your cell membranes and it's
Yeah.
still got that disulfide, so it can still act as an antioxidant there.
Yeah. Help me understand here, like I guess biophysically, why it can't be scavenged. Why does endogenously synthesized lipoic acid why does that need to be the source? Is something impeding that supplemental or exogenous lipoic acid from getting all the way into where it would need to get?
Yeah, so we actually think it's because there isn't an enzyme that can take that lipoic acid and hook it to a protein.
Mm,
And so for the cases where lipoic acid is acting as an enzyme cofactor, it's actually covalently bound to a protein. And the way that the mito fatty acid synthesis pathway works, I said that those fats get synthesized on a carrier protein called the
hmm.
acyl carrier protein. And then once you have your 8 carbon fat made, it gets transferred directly from that acyl carrier protein to target proteins and modified into the lipoic acid so the disulfide gets added. But it's never just free. The enzymes that take it are transferases, so they take it from the acyl carrier protein to the target proteins without ever having a free intermediate. And we think that they just can't recognize and grab that free intermediate if it's not bound to a protein.
Got it. Got it. So once you understand the details of that whole process, basically yeah, there's no way for a free lipoic acid floating around to be grabbed and then stapled onto the correct protein to act as a cofactor. But the endogenously produced lipoic acid is being made in such a way that it does get attached.
Yeah. So we have cell lines or patient cells from patients with mutations, genetic mutations in this pathway. And if we take those cells and we give them exogenous lipoic acid, we can't rescue protein lipoylation at all.
Got it. Got it. And so we need this lipoic acid. We can only get it from mitochondrial synthesis. Insofar, it's going to act as this enzyme cofactor. Seems to be critical, highly conserved pathway. Everything is saying something important is going on here. So I think a good next question is, one hook into figuring out what some stuff is doing in biology is to remove it or break it and see what goes wrong. What can you start to tell us there when this mitochondrial fatty acid synthesis, the ability to produce lipoic acid, breaks down? Maybe you delete a gene, maybe you disrupt something. You can tell us how this is done. What starts to go wrong?
Yeah, absolutely. So we've done this a lot. We've knocked out different steps in the pathway. We often use CRISPR in the lab to make mutations in specific genes. And you lose the first thing you lose, the most obvious thing is that protein lipoylation. But what was unexpected when I was a postdoc was we noticed that when you knock out this pathway, you also start to lose those electron transport chain complexes. So you
Hmm.
have really, really striking reductions in complexes 1, 2, and 4
Hmm.
of the electron transport chain. And what was even more confusing was it didn't seem to be related to lipoic acid at all. So I mentioned we can knock out different steps. And so if we knocked out later steps in the pathway, like the one that adds that disulfide to the lipoic acid, we didn't see the same thing. In those cases where it was only lipoic acid that was affected, the ETC was fine. But if we knocked out actually making those fats on the acyl carrier protein, we lose ETC complexes and we lose the ability of the mitochondria to respire, consume
Hmm.
oxygen.
So something about the process by which this fatty acid is synthesized is important for the electron transport chain to get assembled and function in the way that it needs to.
Yeah. So what we went on to find was that there's actually other products of the pathway that are really important. So again, really early studies before they discovered lipoic acid had shown that this pathway can make longer fatty acids out to 14 or 16 carbons. And once they discovered lipoic acid, it kind of went by the wayside, that observation. And so what we were able to show is that we think it's those longer fats that are important for ETC function. And the reason is because that acyl carrier protein actually physically interacts, it actually physically touches a bunch of different electron transport chain assembly factors. So, what those proteins are, assembly factors, are proteins that are going to help build a functional ETC. So I mentioned that those ETC complexes, they're these really big molecular machines. Like I think complex 1 has somewhere on the order of 45 subunits that have to get assembled all in the right order, in the right stoichiometry, in the right place. And so you have a lot of auxiliary proteins that will help that process. They kind of act like chaperones, if you're
Right.
familiar with that.
Yeah, but I think
They'll,
so. An important thing maybe to understand here for people is when we talk about the proteins in the electron transport chain, or in many other aspects of biology for that matter, it's not like you've got one protein and that's complex 1 and one protein that's complex 2. This is like a whole apparatus of proteins that get stuck together, sort of like, when you build Legos as a kid. To build the whole house, you need to build clusters of Lego blocks kind of in the right order, and all of that needs to be assembled in order to get the thing working properly.
yeah, absolutely. So I talked about how, in addition to those protein subunits, too, the ETC complexes have these really reactive metal cofactors,
Right.
things like iron sulfur clusters and heme and reactive copper. And so often these assembly factors will kind of help hold on to those reactive metals until they're fully assembled in the complex so that they don't like create a bunch of bad reactive oxygen species or other deleterious outcomes for the cell. And so, yeah, it's this whole orchestrated process of building these big complexes in the right order, in the right way, and to get them all fully put together.
Mm-hmm, mm-hmm. So I would imagine. So if the consequence of disrupting fatty acid synthesis in the mitochondria is that the electron transport chain is just screwy, it's not working properly, I would imagine that disrupting this in all cells, like doing a full knockout across all tissues, I mean, that's either going to kill the organism or it's just going to be things are just not going to work all over the place. Is it basically a catastrophic thing to disrupt at the whole body level?
Yeah, total knockout is embryonic lethal in every animal that has to respire. So anything that requires oxygen for life, it can't survive without this pathway. Yeast can survive knocking out this pathway, but they have to do fermentation all the time then.
So, I think it could be interesting to talk about what happens when we do conditional and tissue specific knockouts because you might probably preserve the whole animal and yet like one organ can start to act funny, and that can, that's important for understanding the stuff that you're studying with respect to what this fatty acid synthesis pathway is doing. But I think it's also going to tell us something about like metabolic health more generally in specific tissues. So you can pick the tissue, but I would love to talk about maybe adipose tissue or liver tissue. What happens in either of those two places, or another one that might be a favorite of yours, when you disrupt mitochondrial fatty acid synthesis just in one of those organs?
Yeah. So my lab is working on this in muscle a little bit, um, but we aren't as far along as some of the other studies that have been done in the literature. And so um, just looking at knockout in, say, the liver, people have shown that knocking out the mitoFAS enzyme MECR leads to altered glucose homeostasis. So just in the liver, it leads to this um, really whole body effect of not being able to handle and break down glucose properly. And actually, there's been some similar results with knocking out the enzyme HTD2 in adipose tissue, leading to insulin resistant phenotypes. And so we really think that knocking out this pathway has really large scale effects on your ability to metabolize glucose and
Mhm.
handle it in a whole body
Mhm.
scenario.
Mhm. So another way of saying this is it looks, from what I'm seeing and what I'm hearing from you here, the details might differ somewhat from tissue to tissue, but we could be talking about skeletal muscle. We could be talking about the liver. When you disrupt mitochondrial fatty acid synthesis, you get things like altered glucose homeostasis and insulin resistance. In other words, you're messing with cells' ability to metabolize glucose, even though you're not getting there by overloading them with sugar. You're getting there by disrupting fatty acid synthesis.
Yeah, absolutely. So I think, impairing the ability to break down glucose in two ways. So we talked about that big finding that you really just can't build a functional ETC, an electron transport chain, but it's also related to that canonical product, that lipoic acid as well, because some of the enzymes that use lipoic acid as a cofactor are really important glucose metabolism enzymes. So the most prominent one is pyruvate dehydrogenase, and that's the enzyme that we talked about earlier that is going to make acetyl-CoA from pyruvate.
Hmm. Hmm. What are there human disorders where, I mean, you said this is a lethal mutation when it's like totally like the whole thing is disrupted and so the embryo is just not viable. Are there disorders where people have compromised but still somewhat functional mitochondrial fatty acid synthesis, either across the board or in particular tissues, anything like that?
Yeah, there are patients identified. It's very rare, although the number of patients has been increasing every year, especially with whole exome sequencing becoming more and more available to patients. But there are about 30 patients worldwide with variants that are pathogenic in the mitoFAS gene MECR that I just mentioned. And then there are a few more that have been described with mutations in other pathway enzymes as well. And those patients really look like patients with a number of other primary mitochondrial diseases like Lee syndrome. And so those are typically characterized by neurological problems. So our nerves are really, really dependent on mitochondria for ATP generation. And that's because, sending electrical signals through your neurons all day long requires a lot of energy. And so often when you have patients with genetic disease in mitochondrial gene, you see problems with their neurological systems first. And that's what we see with patients with these variants as well. The syndrome that they get is called MEPAN syndrome, MEPAN. It stands for yeah, I won't even tell you. It's a long enzyme related name, but it's a neurodegenerative disorder.
Hmm.
And it's typified mostly by problems with balance, motor neuron activity. So the patients often have tremor or gait issues, or might even be confined to a wheelchair. Sometimes they have difficulty with speaking or swallowing. And then some patients also develop optic neuropathy as well. So trouble with the optic nerve and
Hmm.
vision problems.
Hmm. And then, so we've talked about, you can probe what something does in biology by breaking it and seeing what goes wrong. You can also look at the other side of the functional equation here. So, it looks like from this paper I'm looking at that some interesting things happen when you, not when you break things in the mitochondrial fatty acid synthesis pathway, but when you sort of supercharge it or overexpress certain components. So, and some of them look like they're actually positive from a health benefit standpoint. What starts to happen when you do those sorts of things? You overexpress components of this mitochondrial synthesis pathway. And so things aren't broken and missing. You're actually getting, I would imagine, more of this fatty acid synthesis.
Yeah, so there's only one or two studies that have looked at this, but they overexpress that acyl carrier protein, so that scaffold that the fats get built on. And they actually found that overexpressing that acyl carrier protein was protective against high-fat diet-induced insulin resistance and obesity. And I believe they also showed that in another study, it was cardioprotective and enhanced mitochondrial function. And so cardioprotective, they were looking at cardiac injuries like after a heart attack, and they showed that it was protective in that scenario.
Mm-hmm, mm-hmm. And so, at a very, very high level, what I would start to think about here, just hearing this stuff, is that okay? So, if the synthesis of these fatty acids in the mitochondria is important for the proper assembly and therefore the proper functioning of the electron transport chain, and a lot of stuff is going to break if the cell can't make ATP the proper way, if the electron transport chain isn't functioning properly. Maybe the things that disrupt that, such as certain diets or other factors that can lead to problems for animals, somehow overexpressing these carrier proteins that are part of this fatty acid synthesis pathway within the mitochondria is somehow protecting animals from that. Maybe it's enabling the electron transport chain to be just a little bit more resilient to these insults that would otherwise start to break it.
Yeah, it's a really great observation. We're very, very interested in this. And we're really interested in the question of if you take a step back and rather than overexpressing the carrier protein, can you drive the same effects by looking at the enzymes in the pathway? So, can you
Hmm.
make more products of the pathway rather than more of the carrier protein, kind of like the substrate? And could you then have similar effects? And then can we take a step back and say, rather than needing to overexpress a protein, is there any way we could develop a diet or a therapeutic that could activate the pathway? But right now, really very, very little is known about how pathway activity is regulated on any level, be it transcriptional, post-translational, or metabolic. And so those are all questions that my group is actively pursuing right now with the goal of really trying to understand and figure out how could we potentially take advantage of those things that regulate the pathway in scenarios like this to a protective benefit.
Yeah. Do we know anything about what modulates this fatty acid synthesis pathway, either up or down in a good way or bad way, with respect to what we would call lifestyle factors? So things like exercise or things like certain diets disrupting this pathway or helping it, things like that.
The only thing we know so far is that it seems to be regulated by the transcriptional programs that regulate mitochondrial programs as a whole. So
Mhm.
things like MYC and PGC1 alpha seem to also drive expression of the pathway genes, but they drive a lot of mitochondrial functions
Got it.
transcriptionally. So we don't know any specific interventions that would target this pathway specifically and not mitochondria as a whole.
Yeah, yeah, yeah. So I mean, it kind of makes sense, I guess, to me. Like the mitochondria, there's many of these components to mitochondria, and they all kind of go together, so to speak.
Yeah.
They're not necessarily tunable, independent of one another.
Yeah. And so I think, we know that it's advantageous to drive those things in other scenarios or for other reasons too, where we would want to drive mitochondrial function, drive mitochondrial metabolism. And so maybe mitochondrial fatty acid synthesis is part of the reason why it's beneficial to drive PGC1 alpha signaling and drive mitochondrial biogenesis.
Mm-hmm. And then, so you briefly mentioned earlier that lipoic acid is sold as a supplement. But we mentioned that you have to improve produce it endogenously for it to be used as a cofactor in in the enzymes we were talking about. So the supplemental lipoic acid isn't going to do that part of it. The cells can't just grab free floating lipoic acid, suck it in, and then use it as a cofactor, but it can still be used as an antioxidant. What can you tell us about the antioxidant function of lipoic acid? How is it actually serving that function?
Um, yeah, I'm getting a little nervous that manufacturers are gonna hate me after this podcast airs, but um, it's really just that disulfide bond that we
Mm-hmm.
talked about earlier can work similar to the disulfide bond in glutathione. It can accept some of those rogue electrons that act as oxidants, and so it can kind of neutralize some of the effects of reactive oxygen species and things like that.
Okay, so basically, if a molecule has a disulfide bond like this, it can probably serve as an antioxidant, just because that's just an inherent part of that chemistry.
Yeah.
Interesting. So, what are some of the things that you guys are working on in the lab today in terms of this mitochondrial fatty acid synthesis pathway? What are the things you're working on, slash, what are sort of the next big questions in your mind?
Yeah. So, I already mentioned we're really keen to understand the regulation of this pathway with an eye always towards how could we be activating the pathway that's already there outside of genetic things like overexpressing a carrier protein or an enzyme. Is there a way that we could activate this pathway through some of the endogenous regulation? And so we've been doing a lot of CRISPR screening type experiments or other studies aimed at, altering nutritional conditions for cells in cell culture and looking at do feeding cells specific nutrients in cell culture media change how much or how little the cell is activating the pathway or using those carbon sources to fuel the pathway to try to understand if it matters, like we were talking about before, if they're metabolizing sugars or fats or, even other substrates like ketones, do those all get made into these fats that are made in the mitochondria in the same way, in the same frequency, or does the pathway prefer some nutrient sources over others? Another thing that we are working on is really just trying to still understand what biological context the pathway is important in. And so I mentioned we're working in skeletal muscle. That's partly because of just a observation that happened by happenstance. We work with muscle cells as our primary kind of workhorse model in the lab because they have a lot of mitochondria and they express high levels of the pathway. And so, if you're going to knock out a gene and study its function, you want to start with a cell line that expresses high levels of that gene, because if it already doesn't express it, you're less likely to see an effect of knocking it out. And so, we
Right.
were we were working with these muscle cells in the culture dish, and you can stimulate them to go from growing proliferative muscle cells in the dish to making an actual differentiated contractile myotube. And it's really fun to do that. You can watch them twitch under the microscope because they'll actually develop that contractile machinery that makes them twitch. And what we noticed with the mitoFAS knockout cells was that the cells couldn't actually make those differentiated muscle cells and they couldn't twitch.
Hmm.
And so we're now using mouse models to look into that further and understand, excuse, sorry. We're now using mouse models to look into that further and understand where in that differentiation signaling and process is talking to the mitochondria and how mitochondria are blocking that differentiation if the mitochondria aren't working properly, such as when mitoFAS is impaired.
Mhm, mhm. One of the final questions I want to ask you specifically is, so when these fatty acids like lipoic acid are made by the mitochondria, well, actually, I just realized I didn't ask you this. I would imagine there's a variety of fatty acids that the mitochondria can synthesize. Is that true? And do they mostly seem to be getting used for the mitochondria in the mitochondria, or do some of them also get exported and maybe used elsewhere?
Yeah, we think that they all stay in the mitochondria and we think they all stay covalently attached to
Hmm.
proteins.
Hmm.
So, in every case that we know of, I talked about lipoic acid being covalently attached to proteins earlier. In that electron transport chain assembly function, they stay covalently attached to that acyl carrier protein as well. And so there's no enzyme like in the cytoplasm when you make fatty acids, the fatty acid synthase in the cytoplasm has a thioesterase domain that's going to have the chemistry that you need to cleave a free fatty acid off and let it go off to its destination in the cell. You need that thioesterase chemistry to do that release reaction. And we don't have an enzyme that we know of in the mitochondria that can perform that activity for this pathway. Now, that's not to say that one exists and just hasn't been found yet, but as we know right now,
Mhm.
they actually can't get cleaved off and go just be a normal free fatty acid.
Yeah. So one thing I'm hearing in this whole discussion is these fatty acids that are synthesized within the mitochondria, like lipoic acid, they aren't just sort of free. They're not going to go like float and become a membrane component like other fats might get used elsewhere. They are intimately and literally attached to proteins, especially the proteins, the enzymes that they're acting as cofactors for. So this whole process of synthesizing them is literally bound up with the construction of these proteins, and they're being used in this kind of functional way rather than as like structural components of membranes.
Yeah, they're definitely, as far as we know so far, fats that act as modifications on proteins. And
Yeah.
you can think of them as almost having like a signaling role
Yeah.
because if they get made, then that enzyme gets activated, sort of, because now it has a cofactor in its active site. Or if they get made, then they support ETC assembly function. And if they don't, then the assembly factors aren't stable, aren't active, or the enzyme's not stable, not active. So you can almost think of them as a signal.
Mm-hmm, mm-hmm. Yeah, and it's easy to forget too. When we learn about stuff in school or by reading papers, we have words and concepts and these discrete buckets our minds like to build and use to segregate ideas and stuff. But when we think about proteins, fats, and carbs as these separate things, and in some sense they are, but in terms of the functional stuff in our cells, like the proteins, it's not really just naked proteins. It's complexes of proteins, many of which are painted and studded with fatty acids and sugars that are stapled to them. And it's that whole thing that is doing the stuff in the cell.
Yeah, absolutely.
Are there any final thoughts you want to leave people with on this general subject of fatty acid synthesis in mitochondria, or perhaps how things like this can kind of go ignored or underappreciated in a field like this?
I think I just would love to leave people with the idea that, we talked a little bit about the MEPAN patients. This is a really rare disease, but I think it's really important to study still because even though individually rare diseases are rare, they're actually very common as a whole. So I think the last statistic I heard was something like 1 in 800 people has a mitochondrial disease. And a lot of the time, those diseases, because they're individually so rare, they have no standard of care. The treatments that we do have are really aimed at symptom mitigation and not, any kind of cure because we just don't understand the disease well enough to know what to do. And so I think it's really important to support rare disease research because, I'm a parent, and most of the time these patients are identified as pediatric patients. And the idea of taking my sick child into a clinician and a geneticist, and you fight years sometimes for this genetic diagnosis, and then to be told, we don't know, there's nothing we can do. It just sounds awful. And so, I hope that the public and people will continue to be really supportive of even rare disease research because, hopefully one day we can have answers for those patients.
All right. Well, Sara Nowinski, thank you very much for your time. And this was fascinating stuff.
Thanks.
Visit mindandmatter.substack.com to find all of my content. Hey everyone, I want to take just a minute to tell you about one of my cool new affiliate partners called Seed Oil Scout. Seed Oil Scout has a really cool map feature that helps you find restaurants in your area where you can find seed oil-free options. They also have a really cool product scanner, so you can take out the app and use the camera functionality of your phone to scan products in grocery stores and see what's actually in these things. Do they contain seed oils? Do they contain other things that you want to know about that aren't necessarily on the box? This will help you find places to eat and find foods to eat that actually have what you want in them and help you avoid those that don't. So if you're looking to cut back on your seed oil consumption, or you just want to know more about what's in the foods on the shelves at the grocery stores, download Seed Oil Scout today. If you go through the link in the episode description, click into that and you'll see a link to the Seed Oil Scout app. If you go through that link on my support page, you'll be directly helping the podcast and you'll be able to download this really cool, easy to use app that will help you find healthier food options. So go through the link in the episode description and download Seed Oil Scout today.